Right. Thank you very much for inviting me to come and give a presentation here to you today. So I'm going to talk to you today about a recombinant protein expression system we developed over the last nine years or so now, where we can export recombinant proteins from E. coli and at really quite high yields. So primarily my research in my lab over the last 20 or so years, probably too long for me to think about remembering, has been on the cytoskeleton, and we've been working on molecular motors and tracks with it that you find within a diverse range of eukaryotic cells, so be those yeast or nematodes or humans. So a lot of what we do is cell biology, and where we get nice images like this so we can see the dynamic movement, but also there's an awful lot of things that we need to do is to make a large amount of protein, to look at kinetic reactions, how different molecules move, interact together, look at how motors run along inside on tracks. So over the time, we've developed a number of recombinant protein expression systems. But this last one came about because we were exploring the impact expression of some recombinant proteins had upon the physiology of the bacterial cell. And the protein that we looked at, actually, when we looked at it, so we fluorescently labeled it, we saw that protein, it was exported from the cells in these vesicles. So E. coli, or many bacteria actually, they make natural OMVs, or outer membrane vesicles. But these are discrete from these because actually you can target the protein that goes into them. The vesicles are much larger, and and you can regulate the amount of vesicles and the amount of quantity by regulating the amount of protein that's produced in them. But I'm going to give a brief overview of this system. So first of all, so it was originally identified. Now we've identified a short peptide. So originally, the original protein we looked at was alpha-synuclein. So this is a neuronal protein that it's involved in, well, its precise function isn't actually known, but it's known that it's involved in membrane or vesicle turnover within neurons of cells. So actually the fact that it made these vesicles in the membrane of an E. coli cell wasn't too much of a surprise. Anyway, so we've narrowed it down now to a really nice short peptide within that protein. And if you just express that with the E. coli, you get the production of these vesicles that we can see here. So if we look on the left-hand side, so here we've got the outer membrane has been labeled with a fluorescent marker, and you can see these vesicles that are forming. Here we've got a nice transmitted or TEM image of the vesicle forming from the outside, from the outer membrane of the cell. Here's a and you can see a cross section of an E. coli cell with the vesicles forming. And in D, this black region here, these are electron dense. These are E. coli cells. So what we did in this experiment is we put the E. coli down on a grid. We left them there overnight to grow, and then we imaged them. And we can see here they produce these, an abundance of vesicles. Right, so we can make vesicles, and we can also target specific proteins into them. So in this strain here, we're showing you, we've got a fluorescent GFP, or green protein here. And in magenta or pink, we've got the outer membrane. So we can see it forming these vesicles really quite clearly. And here we've got an immuno-EM image, and we can see that this is against the GFP, the fluorescent protein. You can see it's all localized within the lumen of these vesicles. So we can target any specific protein into these vesicles that get exported. Then if we just take out the media, so we spin down the cells, we look at the vesicles, we can see them by the EM, and we can look at the fluorescence that's inside these vesicles if we take the same sample and look down the fluorescence microscope. So this is really quite a straightforward process. So here's our culture, pre-induction. We induce with a variety, depends on the promoters. The standard one we use in the lab is T7, so we add IPTG. This is after 24 hours of induction. Well, actually, this is an overnight induction, this experiment here. So we can see here we're expressing a fluorescent protein, so it all goes green. Then we centrifuge out the cells, and you can see the media itself, nice and clear and green. And if we run these through the gel, on a gel, an SDS page gel, we can see we get a really decent amount of protein, and it's reasonably pure in this particular example. So how does it work? So the way it thinks is, so the the tag, we call it vesicle nucleating peptide. So you can reduce it down to as small as 11 amino acids, but actually the optimum size is 38 amino acids, as I said. And it forms this amphiphilic helix that interacts with the membrane with E. coli. And once you get a high enough concentration, so this is a model that we think is how it works. Once you get a high enough concentration, they start to form deformation of the membrane, and you get outward curvature. And with the protein gets incorporated into that vesicle that forms, and then it gets released when it reaches a certain size. And then we can see here is a high resolution or super resolution image where we're looking at live cells, and we can see the vesicle formed when it's filled with the media, filled with the protein of interest. So the purification method is really quick. You can do it in 24 hours or so, the whole experiment. But so this is what we do. So we have a culture, grow them overnight. And here, these are examples. These are images from different stages of the experiment with the green fluorescent protein. We can see lots and lots of lovely green fluorescent cells. We can see the occasional vesicle. We spin out the cells, then we're left with the vesicles. Then we can concentrate them on a filter, really quite rapidly now, and we get a concentrated number of vesicles all there, and it's all enriched there. We know it's pretty. Each step is quite rigorous, and actually, okay, there's a number of things I want to describe here. So first of all, if we take some of these samples here from the overnight culture, we put them on an LB plate that has the antibiotic, the colonies form, so the cells are really quite viable. So it's not affecting viability of the bacterial cells. We spin out the cells, we got the vesicles, we put them onto LB media without any antibiotic, so no antibiotic at all, and it's completely clear, the plate, so nothing grows overnight. And if we shine blue light at these cells here, they just glow. So really quite quick pipeline here for the purification of the protein. So green fluorescent proteins all very nice. It's all very pretty for demonstrating it. But so we want to know whether we could use it to apply this technology to a range of different proteins that are actually useful for biotechnology or industry. So here's where we tagged it with different tags or different model therapeutics. So things like DARP, uricase, stephin, growth factor, EPO, even human growth hormone. And so each of them, we saw export of proteins. So here we've just got an example gel for the DARP. So this one here, the uricase here, and for the stephin, which is here. Okay, so that's where we can see in this lane here, pre-induction, post-induction, and we can see the purified protein. So we get export. The other thing that so the shaded region shows export. The thing that we weren't expecting, but we saw with all the proteins, we saw a huge increase in yield. So for an example, let's just look at this DARP here. So a DARPinol, so it's a protein binding protein, has like antibody-like properties. So if you just try and express the DARP in E. coli by itself, you're lucky to get about 50 milligrams of protein per liter of cell. So all of these are in shaking flasks, okay? Just like we do in the lab. If we put our tag on, we can increase that yield straight away to about 1 gram per liter. Okay, so that happens for a majority of the proteins, and actually from many proteins, we can get yields now with current iterations and with the optimized system, well over 3 grams of recombinant protein per liter of shaking flask. So this is shaking flask E. coli. So works really nicely. That's in shaking flask. Can it be applied to bioreactors? So this is where we went on, did some experiments with our industrial collaborators, Fujifilm Diasynth, up in the northeast of England. And we did the experiments there. We were able to induce, so where we looked at pellet and supernatant. Here's the pre-induction, here's the post-induction, and we can see lots of protein there in the supernatant. Now we've got it optimized with work with the guys up in Fujifilm, or the team up in Fujifilm, shall I say. And now they can get all of the protein actually goes into that supernatant by modifying the growth conditions. So this has been a part of an ongoing collaboration with people at Fujifilm. And so this is my postdoc, Karen Baker, and she went up there for a couple of weeks to go and do some experiments on the amber and the bioreactor systems, working with Chris Lennon and other members of the team up there. So it's a good part of a BBSRC funded project that they are co-funding as well. So we can make all this protein, we can get it out into the vesicles, but is the tag itself, like many proteins, like MBP or SOMO, whatever these different tags, if you remove those, it can be that your protein of interest just becomes insoluble. So we did the cleavage experiment where we put a TEV tag between a VMP, and actually we put a neon green tag in there so we can, it helps us make sure that it's cleaved. So the neon green is a nice fluorescent protein. So it's a fluorescent protein of choice in the lab. So, and we can see it gets cleaved, and in the supernatant, we still see the DARP here. Another one, we did the same experiment with uricase. So we can cleave off the VMP tag and the protein remains soluble. So the primary challenge is to get it to fold correctly and get it into the right environment. And that's what our system allows us to overcome. So we've got the vesicles. We've done quite a lot of characterization of the vesicles, but I guess the important one is what proportion of the protein in those vesicles is our one of interest. And here's some gels that we did some mass spec on. And here's one example with DARP, and it's 40% of the protein within the vesicle is the DARP itself. Okay, so it's between 40 and 60% in most cases. But the other proteins that we found in there, there are small numbers. There's like up here, we've got a small, a very, very small quantities of lots of proteins, just a few of them in there, just one or two, they come up 30%. And then we get a few the most abundant proteins you have in the cytosol of an E. coli cell, like chaperones or transcription factors. Okay, so we can purify it. We've got reasonably purified protein at this point here. And for many applications, this is pure enough. So, for proteins, depends what you want to do in the lab, or if you want to actually apply this for in agri-science, that is enough. At the same time, so that's one thing. In addition, so this system also allows you to do really high, rapid high throughput screening. So I've talked about we can do it in shaking flasks. You can use this system. We can also use it in 96 well plates. So here we are, we've got a 96 well plate. We put our culture in there of the E. coli, grow overnight, so we get the vesicles. Then we can spin the filters down, or we can filter them out really quite easily, just using centrifuges, and then we can do different experiments actually on the media here just by bursting the vesicles. And we can look at different expression of different proteins, look at the fluorescence in the vesicles. We can even look at enzymatic reactions, which is what we've got going on here. So here's one example where we're looking at uricase, one of the examples we have. So if we have no IPTG, we get no expression or no enzymatic activity. So this is where we're looking at the breakdown of uric acid. That's what uricase does. So if you just have no expression, you get no breakdown of the uric acid. You add induction of uricase, you get breakdown of the uric acid. That's exactly the same kind of kinetics as if you were to look at purified protein. And then if you add an inhibitor, then you can also see that lack of activities. So you can use it as a really, really simple high throughput screening method, because you don't even have to purify the protein. And you can look at protein-protein interactions, all sorts of cool things. So you can produce proteins, you can up the yield, and you can make interesting things. So what other things, other proteins can we do? What other methods can we use our system for? So first of all, we found that actually there are many proteins, if you want to grow them in anaerobic conditions, and there are many applications you may want to do that. Many of them, they do not express to reasonable yields in E. coli. We found with our system, actually we can. So here's neon green. This, if you grow it in anaerobic conditions, it will not induce. You just do not get meaningful expression. If you put the VMP in front of it, so exactly the same growth conditions, grown in anaerobic conditions, we get really decent yields. Okay, so it works there. Another reason we're using the system actually is something we're taking forward at the moment, is actually using the bioreactors. So we can put multiple proteins into the same vesicle, and we just follow enzymatic reactions within them. So one simple one we've done here is we've got our protein, which has got the TEV site in between it. So we've got VMP with neon green with the DARP, and there's a TEV site between that neon green and the DARP, right? So we co-express it with the TEV, but again with the VMP. So they're both going into the vesicle, and then it gets cleaved. So we can see the DARP getting cleaved. So in the absence of TEV, this is what the protein looks like. Add the TEV, then it gets cleavage. And this is in the vesicles, okay? It's only in the vesicles we're getting that, well, we can see that cleavage as well. It's getting cleaved in the cells as well, but we can certainly cleaved in the media. So we can see bioreactions within these, and we're taking that forward for a variety of different applications, looking at energy production, for example. So we thought, okay, so if we can do these different reactions, we can help grow things in anaerobic conditions. Can it help us grow things that are toxic? Because you think about compartmentalizing that protein. If it's toxic, it's really good to get it out the environment of the cell, get it out the cytosol. So let's see if we can do that. And what's the most toxic protein we can try and express? Well, ridiculous one. Okay, let's try express DNase 1, because that's going to chop up all the DNA or the genome of the E. coli. Will that work? This is an extreme example. We weren't expecting this to work, because if you look at this is a growth curve in the left-hand side. If you have DNase express it by itself, the cells just do not grow. Eventually, they'll start to grow once they kick out the plasmid, or they mutate the plasmid or the promoter, but they just do not grow. If you add the VMP onto it, then it compartmentalizes it into the vesicles within the cells, and you can see the cells grow. And this is reflected, no protein is expressed in the absence of VMP, so no DNA is expressed, but you add the VMP on, and then you get expression. So it helps the expression of certainly some challenging proteins that we've been working with. Now we've used it for a whole range of different proteins, nanobodies, which are normally disulfide, capsids, viral-like particles, viral-like proteins, and even disulfide bond-containing proteins. We've now enhanced the system. We've screened a whole different range of VMPs. We've now identified some that work at a range of temperatures from 25 degrees, actually even down to 20 degrees, and you'll get the vesicles form. And they work at whole range of temperatures, different proteins, and they get increase in yield. So, and with these, we're starting to see, these yields of well over 3 grams per liter in shaking flasks. Okay, that's the proteins we get. So, yeah. So a lot of this, obviously, this was patented back in 2021, 22, and we published this 2023. And we've been taking this forward now. I just wanted to share some examples of some work that we've been doing since then. So in that paper, we described the ability to express an IgG, so etanercept. So this is a therapeutic. It's used as a therapeutic in the treatment of things like Crohn's disease. It's an IgG fusion. So this is the antibody region here. So it folds like an antibody. It has the disulfide bonds. And so it folds the disulfide bonds. So when we extract it using our system, in the absence of DTT, you get these massive complexes. Add DTT, you get the disulfide bonds break, and you can see the monomer forming there. We can see it binds to protein G or protein A in this example here. So it's the antibody half, the IgG half is binding to what we expect it to. And actually we introduced the TEV site here. So just like the previous experiments, will it remain soluble? Will it remain functional when we remove the tag? And it does. And we can see that over here. So it binds to the protein A. So an interesting thing we found about the TEV when we looked at it, how it interacted with cells, is a small proportion of it. So only a small proportion of it was being exported from the cell into vesicles. The majority of it was staying within the cell. And actually, when we looked at these cells by EM or microscopy, I'm showing the EM here, we got these inward forming vesicles. And again, if we did immuno-EM on the gold particles here, we can see we got these inner vesicles on the inside. Okay, so that suggests perhaps either the disulfide bonds or the dimeric protein there. That's enough to form these inward vesicles rather than outward vesicles. So the way we could test this is it's using a different system that we developed in the lab. So the original protein, remember, that we identified all this working with, it was alpha-synuclein. And if you aminoterminally cetylate alpha-synuclein, it forms a dimer. And here's just a SECMAL analysis showing that we've got acetylated is forming the dimer, unacetylated is forming a monomer. And when we looked at so this is our co-expression system we developed quite a few years ago now. It's published, all the reagents are out there and available to use from AdGene, where you can co-express this protein, and it acetylates your protein of interest within the E. coli cell. So, and that's quite interesting because 90% of all of our proteins are acetylated at their amino terminus, but E. coli just can't do it. But with our system, you can now do it. Anyway, back to the main story. So if we now look at our aminoterminally acetylated alpha-synuclein, now we start getting inward vesicles. Okay, and we can see that those vesicles forming, here we've got fluorescently labeled membrane. So unacetylated on the left-hand side, it's almost majority of it is going out. In the right-hand side, the acetylated form, the majority is coming in. So dimeric, that suggests. One final way we tested that is we stuck a leucine zipper behind the VMP. So leucine zippers are, they're like glue. They bind to each other and they form really strong dimers. So here's just where we've purified the protein to show it is forming a dimer on gel filtration, and it is. When we look at the cells, we're forming these lovely vesicles and they're filled with the protein. So it's definitely dimeric protein that's forming these vesicles, inward vesicles. So no acetylation. So if you've got a monomer, it goes outwards. A dimeric protein, it goes inwards. And that's just an EM image or SIM image showing that. And here's a time-lapse movie of watching these vesicles. Actually, they're really quite dynamic within the cell. So very quickly now, so the final part that I want to really talk about now is, so we've got this, we can form these disulfide bonds. We can show with an IgG type protein that we can form them, and we form these inner vesicles. What about more complex and more challenging, more valuable antibodies? Because we've shown we can form nanobodies, fine, single-stranded strains or these IgG fusions, or homodimeric fusions. But what about things that are heterodimeric, like Fabs or MABs? So we quickly expressed here, we've got a Fab. So this is against lysozyme. And behind we've got the heavy chain and the light chain. Between each of them, we put a VMP tag at the front. So they both go into the vesicles. To confirm whether or not they formed a dimer, we use bimolecular fluorescence complementation. That's where you've got half a GFP that you stick onto the heavy chain. The other half of the GFP, you stick behind the light chain. If the heavy chain and light chain come together, the two halves of the GFP come together and it fluoresces. If you're just getting homodimers, it won't fluoresce at all. And when we look at the cells, we're getting fluorescence. It's forming heterodimers in these inner vesicles here. If we look at the inner membranes of these cells, yep, we're getting inward vesicles. Then we make extracts of the cells. We look at the fluorescence. We're getting extracts in the media. We can see it just quite clearly from the extracts. Then we purify the protein on nickel or on protein G directly to show, it indicates it's functional. It shows there's more fluorescence, so it's forming that heterodimer. And then we do all the binding experiments on protein G, and we got both the heavy chain and the light chain. So it binds to protein G. Does it bind its target? So we did immunoprecipitations now with the lysozyme, and yep, here's the lysozyme here's the lysozyme down here at this band here, and it's coming down specifically with the Fab complex. So it's a functional Fab complex. We did the same things with monoclonal antibodies. This one is an monoclonal antibody against GFP, and we get expression of both the heavy chain and the light chain, as you can see here. When we extract them in the presence of DTT, we can see the heavy chain and light chain. If we've run the gels in the absence of DTT, we can see this tetramer form up here. We can use this antibody. So if we use it in something like 1 in 5,000 dilution, we can use it in Western blots. Like we can see here, we're looking at purified GFP, different quantities, and it recognizes it quite happily. These are extracts from E. coli. You can see it recognizes it there. And we can also use the antibody for things like immunofluorescence, as we can see here, just to demonstrate it works. We've made, okay, here's also showing ELISA to show that it's binding to its epitope with the same kind of affinity to the same antibody if it was to be produced in CHO cells. So we've made antibodies against GFP, anti-His, against HER2, and here's one where we've shown it's against an anti-cancer drug, a therapeutic. It's called MUC1. Again, same antibody. We made the same. Here's the heavy chain. Here's the light chain being expressed in E. coli. Here's where we used it for a Western blot against different cell types. So this protein is upregulated in cancer cells, this anti-MUC1 protein, and we can see it's expressed, and we can use it for immunofluorescence in the mammalian cells. So it works for Western blots, immunofluorescence. Oh, here's the IP. Here's an anti-His antibody as well. But again, yeah, really simple. So within 24 hours, overnight culture, smashing your cells up to purified antibody overnight, and you can do it in E. coli cells. So you no longer need the more, tissue culture systems to be able to produce your antibody. All you need is your monoclonal antibody sequence. We're now exploring, current things we're doing is exploring it in different expression systems, different bacteria. We're looking at mammalian cells. Here we've got some examples of HEC cells expressed in the VMP with GFP just there, so we can see these vesicles forming. We're using it for virus-like particles, as we can see here. And quick summary then. So a lot of time is spent in purifying proteins from cracking open the cells, and you purify them on nickels. Our system allows you to overcome that and simplify that downstream processing. It allows you to up the yield significantly. It allows you to produce proteins that you would not otherwise be able to produce in E. coli as well. So all the constructs that we've made have made available to the academic community are out there, and you can get them from Adgene. And these are the people who did the work. So Tara started this work as a master's student, and now she's been postdocing in the lab with other members of the lab. And here are the people that funded the work, and that's it. So thank you very much for your attention.
Thank you, Professor Mulvihill. So, anyone have questions? Wait a minute, please.
Thank you very much. That was fascinating. So I'm interested in the yield increase in the production of the protein. Would you offer some mechanistic insight as to why that happens to a lot of proteins? I mean, I can understand that for challenging proteins or insoluble proteins, basically you know, compartmentalize that away. But for yield increase, is there any other things behind it mechanistically?
Okay. My hypothesis, or my current model, how it works and why you get these increasing yields is, okay, so eventually, when you try and overexpress a protein, eventually the cell just gets jam-packed full of molecules. And eventually in some extreme cases, you'll get inclusion bodies. What our system allows the whole cell to do is, first of all, it's got the VMP, so it gets it away from the ribosome and it's interacting with the membrane that allows it to fold correctly, which is what's happening with the antibodies. Then it will form the vesicle, and then it gets exported. So it's removing the protein from the cell, so the protein isn't getting so stuffed full of protein, so it can start filling that space so you can keep producing the protein. So the way the system works is that it's giving it time to fold correctly on the membrane, which is how we think that the antibodies are working, because it doesn't need an ER like we always thought. That's my undergraduate understanding how antibodies made. Actually, you just need that membrane interaction to bring things together. But we think the way you can get the high yields is you're just getting rid of the overproduction of that protein, so you've got more space. Hopefully that stands up.
Okay. And so I have another question. So, for other production platforms, like it's in mammalian cells and yeast and stuff, are you looking for like a conservation of this particular kind of like a signal peptide, or what do you think? Or is it like just transplantable from E. coli, you paste it in and then it will
It's
do
all
the
going to
same
be
thing?
okay. The way the system works, it seems all very much dependent upon the membrane that's present. Right. So this is our this sequence is first identified from it's a neuronal cell. So it's the membranes and the lipids you find in your brain. It works really nicely within a membrane of E. coli. We've tweaked the sequence so it binds even more to the one with E. coli. So I think things we are doing now, we're doing further development of the actual sequence of it, so it's optimized for different systems like the yeast or the mammalian cells. That's the way we're going with it, rather than
Oh, sorry,
looking at a
I
homology
missed that
one.
part.
Yeah,
All right, sorry. So it's basically a membrane anchor kind of
yeah,
thing.
that's the way it is.
Okay.
Yeah, that's the way
All
it's
right.
involved
Thank
in
you.
there originally. Yeah.
All right. Thank you.
Okay.
One more question, please.
Sure.
Thank you so much for sharing phenomenal work. So I'm quite interested in antibody production in OMV. So just wonder about the localization. Is it like 100% entrapped in OMV or some surfacely display on OMV?
Okay.
Because
So
I think it would say that it's good, like if you can do surface display of antibody on the OMV, so it can be potentially used for a targeted delivery.
absolutely. And so with the antibodies, because it's a disulfide, and you got the dimeric, it's forming inside the cell. So we've got a new system at the moment that we're currently developing, well, we've got it to work now. We'll be patenting and publishing that soon, I guess, is where we can actually export it. We can functionalize the surface of the E. coli recombinantly, and we can get it to present the different proteins on the surface, be they affimers, nanobodies, whatever it is, and we can fill those vesicles full of antibodies. So in terms of, that's more therapeutic use for us. That's what we're thinking about doing. But yes, certainly that one, watch this space. We can do it. We know we can do it, but it's going to take a bit of time for us to publish that. Okay. But with this system that is available, so the current system that's out there, yeah, so it's, you can make the antibodies, but you can't display it on the surface with this system currently.
Thank you for your answer.